So welcome to the biochemistry session, and where we'll be looking at cholesterol. And by the end of this session, you should answer the following questions. Do you know the structure of cholesterol? What are steroids? Can you describe the biosynthetic pathway of cholesterol? And can you discuss also the regulation of the biosynthetic pathway of cholesterol? So welcome all. My name is Wallace Munyere and I'm the Uredactari. So cholesterol. Cholesterol, it is the characteristic steroid alcohol of animal tissue, which performs a number of essential functions in the body. For example, it is a structural component of cell membranes, where it usually modulates the cell membrane fluidity. It maintains the fluidity of the cell membrane. Also, it is a precursor of bile acids, steroid hormones such as testosterone, estradiol. It is also a precursor of vitamin D. So, the cells of the body need a constant supply of this cholesterol because you have seen it is important, especially in the cell membrane. And so the body has come up with a complex series of transport, biosynthetic, and regulatory mechanisms over the time of evolution. And the liver usually play a central role in the regulation of the cholesterol homeostasis. For example, cholesterol enters the hepatic cholesterol pool from a number of sources. So these are the sources of cholesterol in the body. So there is dietary cholesterol. Also, there is the de novo synthesis of cholesterol in the liver, that is by the hepatic tissue and by the extrahepatic tissue. So those are the three sources of cholesterol in your body. So cholesterol is eliminated by the following means. It can be eliminated as unmodified cholesterol in bile. It can also be converted into bile salts. Also, it can be transported to it can, I've said first, it can be eliminated in bile as unmodified cholesterol, also can be converted into bile salts. The third one is that it can also be transported to extrahepatic tissue in form of very low density lipoproteins. Okay. Okay. So in humans, the balance between cholesterol influx and efflux is not precise, which can result in gradual deposition of cholesterol in the tissue. And cholesterol is notorious in that it usually deposits in the endothelial lining of blood vessels, which usually result in the narrowing of the blood vessel lumen, which can lead to very serious vascular diseases. The narrowing of blood vessels due to deposition of cholesterol in the endothelial lining is known as atherosclerosis. Please remember that. So here we have the sources of cholesterol in the hepatic cholesterol pool. We have dietary cholesterol. You also have cholesterol from extrahepatic tissue. Once you can see from the diet, it is usually absorbed in form of chylomicrons. Okay, the child. Also from the extrahepatic tissue, it is transported in form of high density lipoproteins. There is also the de novo synthesis in the liver. How is it eliminated? It can be transported to extrahepatic tissue in form of very low density lipoproteins. It can also be secreted in form of unmodified or free cholesterol in the bile. It can also be converted into bile salts or bile acids. So cholesterol structure. So cholesterol is a very hydrophobic compound consisting of 4 fused hydrocarbon rings, namely A, B, C, D. So these rings are known as the steroid nucleus. Okay, each ring is known as a steroid nucleus. And on top of that, apart from this steroid nucleus, it has an 8-carbon branched hydrocarbon chain attached to carbon number 17 of the D ring. So this branched hydrocarbon chain consisting of 8 carbons, 1, 2, 3, 4, 5, 6, 7 here, 8. Okay. Then it also has a hydroxyl group at carbon number 3 of ring A here, hydroxyl group. It also has a double bond between carbon number 5 and number 6 of ring B here, double bond. So please understand this structure. I urge you to understand. You can be asked to draw this. So what are steroids? Now, steroids with 8 to 10 carbon atoms in the side chain at carbon number 17 and a hydroxyl group at carbon number 3 are usually classified as steroids. So steroids are like steroids. They are like modified steroids. In what ways are they modified? They have 8 to 10 carbon atoms in the side chain at carbon number 17 and a hydroxyl group at carbon number 3. So cholesterol is the major steroid in animal tissue, which usually arises from de novo synthesis and absorption of dietary cholesterol. Now, intestinal uptake of cholesterol is mediated by the Niemann peak C1, like one protein, okay? And that this protein, the Niemann peak C1 like protein, is usually targeted by the drug ezetimibe. It is targeted by the drug ezetimibe, which usually, and by targeting it, it will reduce the absorption of dietary cholesterol. Note, plant cholesterols, which are known as the phytosterols, such as beta-sitosterol, are poorly absorbed by humans. With only they are only about 5% is the one which is absorbed compared to the 40% in cholesterol. Now, after entering into the enterocytes, okay, after the beta-sitosterol has entered the enterocyte, it is usually actively transported back into the intestinal lumen. It is like the body does not want it, so it is transporting it back into the intestinal lumen, and this is done by an efflux transporter known as ABCG5/8. Now, we have defects in that transporter, which will result into a rare condition known as cytosterolemia, okay? Now, because some cholesterol is transported back as well, plant sterols reduce the absorption of dietary cholesterol. Plant cholesterol such as beta-sitosterol. Let me give you a very good source of beta-sitosterol. Avocado. Avocado have beta-sitosterol. So if you don't want your body to absorb much cholesterol, incorporate avocados in your meal. So daily ingestion of plant sterol esters supplied, for example, in spreads, is one of the number of dietary strategies to reduce plasma cholesterol level. I've given you a very good example. Now let's go to cholesterol esters. Now, most plasma cholesterol is in an esterified form. So you can find this is cholesterol here, but at carbon number 3, where the hydroxyl group is, we have a fatty acid there. Okay, so a fatty acid is esterified here. So this structure no longer is a cholesterol, but a cholesterol ester. Okay, and a cholesterol ester is more hydrophobic even than the non-esterified cholesterol or the free cholesterol. Now, cholesterol esters are not found in membranes and are normally present only in low levels in most cells. Now, because of their hydrophobicity, cholesterol and its esters must be transported in association with the protein as a component of a lipoprotein particle, or they can be solubilized by phospholipids and bile salts in the bile. Okay, so let's talk about the biosynthetic pathway of cholesterol. Cholesterol is synthesized. It is synthesized virtually by all tissues in human, but the liver, intestines, adrenal cortex, reproductive tissue, and placenta make the largest contribution to the cholesterol pool. Now, as with fatty acids, All the carbon atoms in cholesterol are provided by the acetyl-CoA and nicotinamide adenine dinucleotide phosphate usually provide the reducing equivalence. Okay, now this pathway is endogonic. It takes up energy. It requires energy, being driven by the hydrolysis of high energy thioester bond of acetyl-CoA and the terminal phosphate bond of ATP. So there is investment of ATP. Now, synthesis requires enzyme in the cytosol. So it is a cytosolic process. Also, there is an enzyme found in the membrane of the smooth endoplasmic reticulum, and also the peroxisomes are required. This pathway is responsive to changes in cholesterol concentration, and the regulatory mechanisms exist to balance the rate of cholesterol synthesis against the rate of cholesterol excretion. Now, when there is an imbalance in this regulation, can lead to elevation in circulating levels of plasma cholesterol, and this might cause atherosclerosis or vascular disease. So, in cholesterol synthesis, you must at first synthesize 3-hydroxy-3-methyl-glutyryl-CoA. So the first two reactions in cholesterol biosynthetic pathway are similar to those in the pathway that produce ketone bodies. I will do a video on the ketone bodies. So they result in the production of 3-hydroxy-3-methyl-glutyryl-CoA, the HMG-CoA. First, the first reaction, two acetyl-CoA molecules condense to form acetyl-CoA. Then next, a third molecule of acetyl-CoA is added by HMG-CoA synthase, producing HMG-CoA, the 3-hydroxy-3-methyl-glutyryl-CoA, which is a 6-carbon compound. Now, in the liver parenchymal cells or the hepatocytes contain two isozymes of the synthase, the HMG-CoA synthase. We have a cytosolic enzyme which will participate in the cholesterol synthesis, and a mitochondrial enzyme which will participate in the ketone body synthesis. Please remember that point. Here we are. These are two. Maybe I should remove this. Or should I move that? Now, this is the structure of acetyl-CoA. The two of them combine, catalyzed by thiolase, to form acetyl-CoA. Then a third acetyl-CoA is added here, catalyzed by the HMG-CoA synthase. We will also put in water. The CoA part will move, will go to the cytosol, and the compound formed here, a 6-carbon compound, 1, 2, 3, 4, 5, 6, okay, known as the 3-hydroxy-3-methyl-glutyryl-CoA, okay, 6 carbons. Then what happens to the 3-hydroxy-3-methyl-glutyryl-CoA? Next, it is reduced to mevalonate by HMG-CoA reductase. Okay, this reaction is the rate limiting and key regulated step in cholesterol synthesis. That is why, please remember that the reaction which is the rate limiting is this reaction, the one catalyzed by the HMG-CoA reductase, and it occurs in the cytosol, uses 2 molecules of NADPH as the reducing agent and reduces CoA, making the reaction irreversible. It is irreversible. Now, HMG-CoA reductase is an integral membrane protein of the smooth endoplasmic reticulum with its catalytic domain projecting to the cytosol. That is why I've said also the membrane of a smooth endoplasmic reticulum participates in cholesterol synthesis, because the HMG-CoA reductase is bound to the membrane of the granular endoplasmic reticulum, or the smooth endoplasmic reticulum. Here you can see your HMG-CoA. Then HMG-CoA reductase will be put into NAD pH, and it will undergo reduction to form mevaronate, which is also 6 carbon. Now, let's continue. What happens to mevaronate? Mevaronate will undergo a series of reactions, a complex series of reactions, which some of you might be interested to know, others might not, but I will go through them, but these are just a series of reactions. You can see here mevaronate 6 carbons will undergo phosphorylation. There will be phosphorylation of mevaronate, and by phosphorylation, so mevaronate is converted to 5. You can see a mevaronate is converted to 5 pyrophosphomevaronate in 2 steps, each of which transfers a phosphate group from ATP. So ATP will undergo phosphorylation, mevaronate to form 5 pyrophosphomevaronate. Then in the second reaction, a 5 carbon here. Now a 5 carbon isoprene unit, in the second reaction, the 5 pyrophosphomevaronate will undergo decarboxylation. There will be also you can see energy is being put in. Okay. Now to form this is a 5 carbon isoprene ring, isoprene unit, not a ring, known as the isopentyl pyrophosphate, IPP. Okay. This is an isoprene unit, a 5 carbon isoprene unit. Okay. Then now this IPP is a precursor of a family of molecules with a diverse function known as the isoprenoids. Okay. And the cholesterol is a sterol isoprenoid. And if you can remember, non-steroid isoprenoids include ubiquinone or your coenzyme Q. Now, those are other details. Then the isopentyl pyrophosphate will undergo isomerization to form 3,3-dimethyl allyl pyrophosphate. This isomerization, so an isomerase. Then this IPP, and let's call this DPP. So the isopentyl pyrophosphate and the dimethyl allyl pyrophosphate will combine. Okay, they condense to form a 10 carbon compound known as the geranyl pyrophosphate. Okay, then a second IPP. When I say IPP, I mean the isopentyl pyrophosphate will condense with the 10 carbon compound, the geranyl pyrophosphate, to form valencyl pyrophosphate, 15 carbons. Okay. Then 2 of these FPP, the valencyl pyrophosphate, will condense to combine, and 15 plus 15 to form a 30 carbon compound known as squalene. Okay. Known as squalene. Okay. So squalene is converted to, now this squalene is converted to lanosterol by a sequence of 2 reactions. Sequence of 2 reactions here, indicated by the 2 arrows, and catalyzed by the squalene monooxygenase, which uses a molecular oxygen, and these enzymes are associated with the smooth endoplasmic reticulum. Once lanosterol is formed, which is the first sterol, it is converted to cholesterol. It is converted to cholesterol by in 8 steps. So lanosterol will undergo 8 steps. Of the steps, there will be shortening of the side chains, oxidative removal of methyl groups, also reduction of the double bonds. There will be also migration of the double bonds. So with these 8 reactions, there are 8 reactions to convert lanosterol, which is the first sterol, to cholesterol. So once cholesterol is formed, now that is the de novo synthesis. It occurs mainly in the liver, but also extrahepatic tissue such as adrenal cortex, the gonads, it will take place there. Now, how is this regulated? Now, HMG-CoA reductase is the major control point of cholesterol biosynthesis and is subject to different kinds of metabolic control. First, we have the steroid-independent phosphorylation and dysphosphorylation. So, HMG-CoA reductase activities is controlled covalently through the actions of adenosine monophosphate activated protein kinase. Okay, so and phosphoprotein phosphatase. Now, the phosphorylated, please understand this, the phosphorylated form of the enzyme is inactive, not active, inactive, whereas the dephosphorylated form is active. So note that the adenosine monophosphate activated protein kinase is activated by adenosine monophosphate. So cholesterol synthesis, like fatty acid synthesis, is decreased when ATP availability is decreased. So when there is a low ATP, so with the cholesterol synthesis below. But when ATP is high, is the other way. So when there is low ATP, the enzyme, the HMG-CoA will be activated. So there is also hormonal regulation. The activity of HMG-CoA reductase is controlled hormonally. An increase in insulin favors dephosphorylation, which is the activation of the reductase, whereas increase in glucagon and epinephrine or your adrenaline has an opposite effect. It will lead to phosphorylation, which is also inactivation of the enzyme. So phosphorylation of the reductase will result in inactivation. There is also drug inhibition. We have statin drugs, the atorvastatin, fluvastatin, lovastatin, pravastatin, and others, which are structural analogs of HMG-CoA. So they are competitive inhibitors of HMG-CoA reductase. And these drugs are usually used to decrease plasma cholesterol level in patients with hypercholesterolemia. This hypercholesterolemia is familial. It can be inherited. So you find that in some families, they will use these drugs to reduce the plasma cholesterol levels. So thank you for your attention, guys, and please subscribe for more anatomy and biochemistry content. Thank you.